And if you have a more pressing question, like a clarification question, I'm pretty bad at multitasking, but I think Danny is going to be monitoring those. So if it needs to be answered right away, hopefully he'll be able to identify that and gently interrupt me. Yeah, so thanks for being here. I'm going to start with a warning that I'm going to start this talk with some bad news. But I don't want you to worry, because later on in the talk, it's going to get more optimistic. So the bad news here is that cognitive ability declines with age. And you've probably heard of the dementias, like Alzheimer's disease, that impair mental abilities. But even in normal aging, in the absence of pathology, we see declines in cognitive ability across a number of cognitive domains. So this figure shows the typical cross-sectional pattern that is observed. And I'm going to get a little pointer here. Okay, hopefully you can see my pointer. This T-score thing, this is just a way to standardize scores across different items so that we can use the same y-axis for all of these different types of cognitive domains. And just as a point of clarification, and this is mostly for people who are more used to using stats, these T-scores are different from the more common T-statistic. So this is called a psychometric T-score. It's kind of confusing. It's a specialized term, but what you need to know about it is that the average T-score is 50 for an item, and the standard deviation is 10. And what you usually see in these cross-sectional figures is some separation between cognitive domains that are considered part of crystallized intelligence. So these things such as verbal ability and numerical ability. These are supposed to reflect the accumulation of knowledge and skills that you've learned and practiced throughout life. And then there are those that are considered to be fluid intelligence abilities that are all kind of lumped in this linear decline, which reflect the ability to reason and think flexibly, often about new problems. And fluid intelligence tends to decline more with age, as you can see here, whereas the crystallized intelligence seems to stay stable or even increase over the lifespan. Okay, so interestingly, this is the more common graph that you'll see. But interestingly, if you look at longitudinal trajectories where you follow an individual across time, you usually see a much more optimistic picture. So perceptual speed, that's in green here, still seems to decline pretty linearly across the lifespan. But for many other domains, there seems to be good stability on average until around the mid-60s. And either way, in both of these pictures, though, we observe that later in life, cognitive abilities tend to decline. So before I go on with the talk, I thought it would be fun to kind of warm everyone up, introduce you to some of the types of tasks that make up these cognitive domains so that you have some sense of what we're talking about here. So I'm going to show or describe some example tasks, but there are many different types of tasks that try to get at these different constructs that we're interested in, these things: inductive reasoning, spatial orientation, etc. So I'm going to start with inductive reasoning here. So this is an example of a figural inductive reasoning task. These matrices have to be completed by one of the given answer alternatives on the bottom here. And in order to find the correct answer alternative, you need to infer the rule or rules which govern the variation of the elements of the matrix here. So I'm going to put up a poll here to see what people think the answer is to this one. And I'll give you about 30 seconds to respond. Often these tests are timed, but more importantly, my talk is timed. So I'll have to cut you off. And let me launch this poll. Okay, so you have about 30 seconds. All right, so if you haven't voted, time to put in your answers. I'm going to end it. Okay, so you should be seeing the results now. The majority of people chose the correct answer in this case, which was B. And if you said B, you're correct. Okay. So now we're going to move on to a little bit of a trickier one now that we're warmed up. So this one's similar to the last example, except now the inferred rules can be applied either horizontally or vertically. So I'm going to put up a poll again where you can vote now. The answers that you can vote between are over here on the right, and then you're trying to solve for this box. Okay. So let's see. And I'll give you a minute for this one because it's a little bit trickier. All right, I'll give you a few seconds to put in your votes if you haven't voted. Just guess for whatever you're feeling, sort of. And I'll close it up in a few seconds here. Still coming in. Anyone else? Any last guesses? Okay, I'll end this. So again, you guys did great. You got the correct answer here, which was C. If you didn't get it, I know these puzzles can be a bit addicting, so I'm not going to go over why the answer is C, and I'll put it up again at the end for you, if in case anyone wants to work on it more. Or you could go to, wait, where's my laser pointer? To this paper, which is where the example is located, but I don't think they explain how you get to the answer there either. Okay, so here's another type of task that assesses spatial orientation. Oh, did I close the poll? I'm not sure if I shared them, so I'll share them for a second, and then I'll stop sharing them. Okay. So here's another type of task that assesses spatial orientation. And in this task, you look at this target on the left, and then you determine which answer or answers on the right match the target. So for this one, there can be more than one correct answer. And I'll give you 30 seconds to work through this one too. All right, give you a few more seconds to get your final votes in. And I'll end it and share. So here's what you guys thought. The answers are actually A and D here. So if you guess both A and D, that's the perfect answer. Looks like there were some differences between A and D. Okay, so you can take a breath now. I'm done quizzing you for now. I'm just going to explain some of the other of the tasks. And good job as a group. You guys did really well. So this is what a perceptual speed task might look like. I might give you something like this. In this task, you have to indicate as quickly as you can whether any of these 5 symbols to the right match any of the two symbols, either of the two symbols to the left. So it can be either or, one or the other. And you would just say yes, no, yes, yes, like as fast as you could. Usually you'd have a longer list, something like that, where you're really trying to maybe compare items very quickly. And then I'm just going back here so that we can remember the other tasks. For numeric ability, it would be something like simple arithmetic problems, usually with a timed component there too. Verbal ability is usually some sort of vocabulary tests. And verbal memory is usually a task like, I'll tell you a story and have you repeat as much back to me as you can remember. Yeah, so as you can imagine, if you do enough of these tests in a row, it can be pretty tiring. I also just wanted to mention that sometimes interpretation of tests like these can be limiting because we're usually trying to find cognitive constructs that everyone that we're testing have in common. So sort of by definition, this makes this idea of crystallized intelligence, which is based on learned knowledge and practiced patterns, it makes it pretty difficult to assess generally and limited to very specific cases, like usually something like vocabulary or arithmetic that we really expect everyone to share in common. And I think that this sometimes has the result of underplaying the importance of crystallized intelligence. But if you think of how much specific knowledge is needed to do most jobs in daily life well and efficiently, you realize how important it is. So for example, a teenager might be able to learn some aspects of law faster than a 60-year-old if neither of them has been exposed to it before. But someone who has been practicing law for 30 years would be much more equipped to actually practice law than the teenager in the real world, because to do it well, you need to know a lot. And in fact, well, I mean, I don't really know anything about law, so I don't know this for sure, but I would predict that the 60-year-old lawyer would even learn new law, much faster than the teenager, because they've spent decades developing efficient representations about the structure of law. So yeah, I'm starting to add lots of hope and optimism here. So let's go back to more depressing stuff. And that's that a lot of the negative patterns that we see in cognitive ability are also reflected in measures of brain health. This applies to many metrics of brain health, but I just picked an intuitive one to think about, the total brain volume. So in this figure, the red, oops, I lost my laser pointer. Here we go. The red is the females and the blue are males, which isn't important at all to what I'm trying to show you, but I just took this figure from this paper by Liu et al. So I left the colors as they were. But I picked this paper mainly because it shows longitudinal changes. So that's what these lines are between connected points. So each line represents an individual's first MRI and second MRI. And I want you to focus on a couple of points. First, that overall brain volumes tend to decrease with age. And this seems to accelerate with older age, but also that there's a lot of inter-individual variability. So like if you look at some of these folks, their brain volume is actually increasing. This one has huge increases. So there's a lot of flexibility here. And this is really one of the messages that I want people to take home after the talk, that you shouldn't feel limited by these sort of average patterns when you hear about these things in the popular press. There's actually a lot of individual variability in this area. In the same vein, often the variance itself reflects really smaller differences than I think people expect or intuit a lot of the time. So here I'm showing an image from a study by Ledig et al. And I wanted to show this image for a few reasons. One reason is just to show you some brains, but also I wanted to emphasize that it's often hard to say anything about an individual's cognitive ability just by looking at their brains. So here I'm showing, or they're showing rather, a healthy control up here on this row, a patient with mild cognitive impairment who later transitions into Alzheimer's disease over the course of the study. And also a patient with Alzheimer's disease that started with Alzheimer's. And then in the columns, they're showing images like a baseline image, an image taken 2 years after that baseline image. And in the third column, they're highlighting where there's differences between the two images in these blue and red colors. So blue represents decreases in volume and red represents increases in volume. So the first point is that it's very hard to see striking differences between the healthy control subject and the patient with mild cognitive impairment, even though there's pretty substantial differences in cognitive ability between these two individuals. And in the same way, it's hard to tell in an individual how old they are just by looking at their brain. You can see some of visible differences with their choice of the Alzheimer's disease patient. There's more atrophy near the ventricles, these areas here. And in the structure called the hippocampus, there's more atrophy down here. But in this paper, they actually say in their own words, many studies have shown that biomarkers of morphometry, such as volume or shape, correlate with Alzheimer's disease progression. However, intersubject variability can lead to substantial overlap with the healthy population and thus limit the discriminative power of these features. So not all Alzheimer's patients look like this. And finally, you can see that in all subjects, there is a pretty subtle differences in their images, even over 2 years. Like these changes are really small and just on the border of some of the areas that we're looking at. Okay, because I work a lot with some of these high-tech measuring devices like MRI, I myself have had the unique and somewhat depressing opportunity to observe my own brain atrophy over my research training period. So I'm highlighting in red where I observe volumetric loss in my own brain. So it's a nice personal motivator to do the work that I'm doing and kind of egg me on. Another interesting thing is that there are regional differences in the brain where these changes happen. So on the left, I'm showing a map from one of the studies from our lab, showing where volumetric changes with age are the most significant. So those are these areas that are highlighted in blue. And on the right, I'm showing longitudinal plots again, the same way that I showed you before, these connected dots of the same subject over time. So this is from a study from another lab, which shows volumetric relationships with age in the lateral prefrontal cortex here, which is an area that reliably shows more volumetric decline with age, and the primary visual cortex, which usually doesn't show much change with age and didn't in this study either. And again, you can see that not everyone has negative changes over the 2 time points. There's some individuals that increase. And also that there's large intersubject variability. For instance, in the lateral prefrontal cortex, we have this 80-year-old here with the largest volume of the 80-year-olds is about the same as the smallest volume in the 25-year-olds in this study. So we can observe these average trends, but it's really hard to be very confident about any individual. Okay. We also see that measures that are thought to be somewhat reflective of vascular health decrease with age. So such as cerebral perfusion is a measure that I've worked on. I'm showing data here from our lab, but other labs have shown this pattern too, where the cerebral blood flow decreases with age. And again, this seems to be particularly true in certain regions of the brain. I used pretty large regions here, but this is just showing that in this broad area of the parietal lobe, I see slightly more reliable decline with age than if I look at the whole brain altogether, although I do observe this pattern globally. So part of what I wanted to do here is give you all some sense of the methods that we use along with this sort of overarching narrative. And so I wanted to start by talking about how to get that perfusion data. We use a method called arterial spin labeling, which we can do with magnetic resonance imaging. So to start, we take some volunteer, and of course we need to protect their identity so that no one can tell who it is. And we put them in an MRI scanner. And then In this technique, we can magnetically label water completely non-invasively to create a sort of intrinsic contrast agent. So we can label the blood, usually somewhere at the base of the head, and then we will wait a second. After we label it, we'll wait a second or two for the blood that was down there to travel up into the brain. So we know that the arterial blood is traveling upwards here because all of the blood that goes up to the brain is going through just a few arteries in the neck. And then we can acquire an image of the brain after letting the blood get there. And since the water in the blood was magnetically labeled, its contribution to the overall signal in the image is suppressed. So then we do the same thing again, except this time we don't label the blood and acquire another image where its contribution to the signal is not suppressed. So we have these pairs of tagged images and untagged or control images. And since the only difference between the signal in the two images is from the presence or absence of water from down here, the difference between the tag and control image is proportional to the amount of perfusion. So we can use what we know about physiology and physics to try to be more quantitative. That's what like this equation's doing. And you'll usually see this quantification in some exact units then, like milliliters per 100 grams of tissue per minute. This is a somewhat scary equation for a general talk, but this just boils down to some expectations that we have about the physiology and physics. So let's just simplify it a little bit. Just to say that if we apply some expectations about physiology, we can make the technique quantitative. And after doing that, we get these beautiful images that are brighter, where there's more perfusion, usually on the outside in the cortex, and darker where there's less perfusion. So you can see in this example, for instance, that the gray matter of the cortex, where most of the neuronal cell bodies are, the perfusion is higher. Well, in the white matter, that's more inside here, it has more of the connections and supporting cells in the brain, but less of the neurons itself. And those areas have less perfusion. So I can just like average over these little pixels, or if they're volumetric, we call them voxels. And then that's how I get those measures that I showed you earlier. Okay, so where are we? Seems like mental ability declines with age, brain health seems to decline with age, vascular health seems to decline with age. So this all seems pretty depressing. But there's good news. And the good news is that it seems like certain lifestyle factors can really influence this trajectory of cognitive decline. And one of the most promising factors is engaging in light to moderate aerobic exercise. So this figure was quite impactful in cognitive neuroscience and was from a meta-analysis done in Art Kramer's lab while he was at the University of Illinois. They looked at all the randomized control trials on exercise interventions for sedentary older adults that existed at the time that had cognitive components. And they found that aerobic exercise interventions had actually really impressive effects at improving cognitive performance, and particularly on these so-called executive functioning tasks. So these include things like working memory and mental flexibility components. But really all of these cognitive domains that they're looking at here would be considered more in the fluid intelligence category that we talked about earlier. So the control groups here in blue were usually put into like some kind of stretching, active stretching intervention, so that they still had something to do, like some kind of active intervention, but it wasn't aerobic in any way. So this advice to do aerobic exercise for your cognitive health, it's been around for a while now. So I think that many people know this or have heard of it. And to some degree, just knowing this in itself is really helpful. But the observation was a bit odd. Even if it doesn't seem that way anymore, it's like, why would doing light to moderate aerobic exercise make you smarter? So this question It's still a very active area of research, because if we can understand the physiological mechanisms behind these benefits a little bit better, then maybe we can design more effective interventions or pharmaceuticals in the future. Okay, so here we have this list of observations again. And in our lab, we started hypothesizing that perhaps one of the ways that aerobic exercise helps with age-related cognitive decline was by improving cerebral vascular health in some way. So a healthy vascular system in the brain, it might improve overall brain health and function, which then would improve mental abilities. And I'll say as a caveat here that exercise has other effects on physiology too, not just on vascular health. So we weren't hypothesizing that this is the exclusive way that exercise might help, but just that it might be one important factor. So one of the first studies that I was part of was trying to establish the relationship between cardiorespiratory fitness and age-related changes in cerebral perfusion, or what I call here cerebral blood flow. I just use those terms synonymously a lot, the CBF. So what I'm showing here is a graphical representation of a mediation analysis. The top part shows that in our sample of older adults, age negatively predicted cerebral blood flow. So this R is a measure of the size of the correlation. You can see that it's negative here. And this p-value is a measure of the statistical significance. So lower is better. Down here, first, this just shows that age also predicts lower cardiorespiratory fitness. And then we see what happens when we try to predict cerebral blood flow by age and an estimate of cardiorespiratory fitness at the same time. And when we do that, we see that cardiorespiratory fitness still significantly predicts cerebral blood flow. That's what this is showing over here, even when we control for the effects of age. That's what this is. But on the other hand, the ability for age alone to predict cerebral blood flow substantially decreases when you control for the effect of fitness. And in our sample, this relationship was no longer significant after doing that. So we can interpret this as saying that a lot of the age-related decreases that are typically observed in cerebral blood flow can actually be explained pretty well by decreases in cardiorespiratory fitness. And that's very highly related to level of activity. So since then, our lab has taken this research much farther and has developed a working framework that describes a cascade of effects that help explain age-related cognitive decline. And if you're interested, I encourage you to check out this paper down here. I have a little reference listed. It's in Network Neuroscience, which describes this framework and some of the evidence that points to it. So this framework shows a cascade where age-related cerebrovascular decline propagates to structural decline, which propagates to functional decline, and eventually manifests as cognitive impairment. And our lab does research on all the levels of this proposed cascade model, but I'm going to be focusing in on the cerebrovascular health declines because I particularly wanted to introduce a method that was invented in our lab that I think is pretty cool. But before I show you that, I wanted to sort of zoom in on that cerebrovascular decline component. And if we do that, we see that actually cerebrovascular dysfunction comprises a number of both structural and functional factors. So these are the more structural factors. These are the more functional factors. And these functional and structural changes are what are thought to actually contribute more to the damage in the brain and the cognitive impairment. But we see that arterial stiffening upstream, this often appears to start occurring before these downstream changes that seem to be more damaging. And I'll just plug now that I have a fairly comprehensive review coming out on this topic and not too long. So I encourage you to read that if you're interested in the details of this topic. I'll include my email address at the end of the talk, so you can let me know if you want me to share that article with you when it's published. In reality, things are more complex, and all of these factors interact with each other and exacerbate earlier effects. But the idea with this more simplified model is just to show that if things can be done to prevent or postpone arterial stiffening, that is a good level to want to measure and intervene on to predict and prevent these downstream effects. So in our lab, we've been developing a method based on diffuse optical imaging that helps non-invasively measure the stiffness of the cerebral arteries. So I'll introduce that method here. This method works by shining near-infrared light into the head, which passes through the skull into the brain, and then detecting it at some distance away. So you can imagine that different photons from the source of light here have different fates. Some of them will bounce around the tissue, and as luck would have it, they end up bouncing back into the detector. So those are the ones that we end up seeing and that we can measure. A lot of the other photons we never see. They just fly out of the brain, like this one. And actually, this is good because it helps us guess where in the brain a photon was likely to bounce through if it ended up reaching the detector. So like, for instance, a photon that starts bouncing around here, it's unlikely that a photon that passed around here will reach the detector because it's so much more likely to fly out of the skull if it's near the edge of the skull. And finally, there's some molecules in the tissue, especially oxygenated hemoglobin, which can absorb the photons. So that's like what that is. And that's the sound it makes when it absorbs light. So here's the key point. If there is more oxygenated hemoglobin, it absorbs more of the light. So then less of the light will end up reaching the detector if there's more of this here. And this actually happens very strongly in the arteries with every heartbeat. So after each heartbeat, the amount of light that reaches the detector drops when that pulse wave reaches the area that you're measuring from. So this pulse wave is actually a relatively large signal that we can often measure on single trials. And for a lot of other applications in the lab where we're interested in smaller signals, this is kind of a big problem. And lab members have spent a lot of time and effort trying to figure out how to remove this signal well. And what I think was some really impressive creativity, my advisors saw the potential to use this, what we usually considered noise, for a really useful application. So the idea was that perhaps we could infer some attributes about how stiff the arteries were or how healthy the arteries were by looking at parameters and the shape of this pulse wave. And I'll talk about that more in a second. But before going into the details of that, I wanted to emphasize that why we're doing this, like one of the main advantages of this technology is that right now it's really the only tool to measure local differences in arterial stiffness non-invasively over the whole head. So like, this is what our helmets look like, where we put optical fibers in detectors. We can put optical fibers in these smaller tubes and detectors in these bigger tubes, and we can cover the whole head at the same time. This is just showing mapped onto an MRI image of the scalp, where the source locations and detector locations end up being. So here's the prediction. In younger or healthier arteries, there's more elasticity in the arterial wall. So that elasticity helps absorb some of the pressure from the pulse wave, and the shape of that pulse wave then becomes more elongated over time and not as peaky because it's sort of stretching and then pushing back. For the older or less healthy arteries, the arterial wall, it can't absorb as much pressure because it's lost some of that elasticity. So the pulse wave becomes more pulsatile in its shape. So there's other dimensions besides the shape that we can look at. For instance, you can just look at the amplitude. How much change in light do we see? It turns out that on average, this is mostly related to the blood pressure. We can also look at the transit time, which reflects how long the pulse wave traveled to reach the area that you're measuring from. So this would be a longer amount of time than this one, which got there right away. And we can also derive metrics that try to capture the shape in some way, like the difference between the peakiness here or this drawn out response. And we've published numerous papers now looking at all of these metrics, and the pulse shape, this measure here, seems to be the most reflective of arterial stiffness. So that's what I'll focus on for the last part of the talk here. So this is a figure from one of our lab's papers showing that our measure of arterial compliance, that's on the y-axis here, this is just the opposite of stiffness. So it's the same measure, but it's just called differently. It declines with age. And interestingly, if we look over the whole brain, so this is like a regional map of the brain. These are sagittal views, and this is from on top. It looks like there's more stiffening in certain areas of the brain. So this red is more stiff and blue is more compliant. And you can see that it tends to be more, we tend to observe more stiffness in these frontal areas. Arterial compliance, we've also observed that this measure predicts brain volumes. So I'm showing here that it predicts cortical gray matter volume, white matter volume here, and subcortical gray matter volume. So this is really all over the brain. It looks like the prediction is the strongest for white matter volume. And this makes sense to me in some ways because overall the white matter is further downstream from the arterial tree. So we might expect that vascular dysfunction affects these regions first. There's another MRI measure of white matter health, which is the load of white matter signal abnormalities, or WMSAs. These reflect small lesions in the white matter of the brain that are thought to be indicative of microvascular damage. And we see that the higher your arterial compliance is, the lower number of these white matter abnormalities we see as well. And finally, I just want to point out that there seems to be regional specificity of arterial compliance in terms of predicting cognitive ability. For instance, starting in the middle here, if we look at the arterial compliance in the frontoparietal areas of the brain, so that those are these areas that are outlined here, these areas are where we typically see more structural decline with age. We also see more decline in arterial compliance with age. And the arterial compliance here also predicts a cognitive task. So that's what we're going back up here to A, the same regions, but it predicts a task that is thought to be functionally associated with these frontoparietal areas. So this is called the O-span or operation span task. And it has you memorizing sequences, but then doing math problems between being tested on the sequences. So you're trying to sort of hold a lot of information at once. But then down here, I'm showing that in the visual cortex, remember, this is an area where we don't see structural decline usually. It seems to be pretty preserved with age. We see the same relationship with arterial compliance again, that it doesn't really change that much with age. So we think that the ability to see specific regions with this method is actually going to be really useful. And we're starting to explore now in a more longitudinal way. All of these that I've shown so far are cross-sectional, whether these metrics predict future changes to brain and cognitive health. Okay, so distilling all of this down into some take-home messages that I'm hoping that you leave with a couple of core ideas. So first is that aerobic exercise is good for your cognitive health, and really even light to moderate intensity exercise like walking at a good clip seems to do the job. You don't need to become a marathon runner to benefit. Secondly, I want to leave you with some sense that these age-related declines in cognition and brain health that we see on average does not define an individual's fate. So if I knew your age and I saw your brain and I had all this information, I wouldn't be able to do a very good job predicting your cognitive performance on these tests. And these are even on tests that are supposed to assess these more general constructs. And I would completely fail at predicting more specific skills like doing law or programming or something like that. And finally, I hope you leave with some idea of the types of measures that we're developing and kind of like how cool the lab I'm in is, because I'm really proud of it and the work that we do there. So here's our cool lab. The lab is actually a bit different in composition now, and this is an older picture, but I wanted to highlight a lot of the individuals who are instrumental in the projects that I was presenting here. So I guess I'll come back here, but then I just wanted to show now a more relevant picture of who's currently in the lab and carrying on this research. And again, I'll just leave my email here and also the website if you're interested in seeing what the lab is up to or enrolling to participate in any studies. You can go to this address. And yeah, I'm ready to take some questions now if you have any. So I don't know if I should stop sharing or like put up the inductive reasoning task. I guess let's stop sharing for the questions and then I'll put it back up at the end.
All right. So yeah, we got a few questions. One of the first questions we had was, what factors might lead to an individual's brain size increasing with age, as you saw in some of those longitudinal studies?
Yeah, so there's some ideas about this. Some people have observed that as you train certain types of tasks, the brain tissue actually grows. Like I think that one of the first ones that I saw about this with like skill-based learning was if you do like juggling tasks, you'll see some specific regional growth, and that might be reflective of the tissue. Usually it's not as much cells, like new cells being formed, but more density of synapses becoming greater. But also we think that some of those volumetric changes are also reflective of the vasculature itself. So as there's more capillary growth in those areas, that can also add to the volume.
Great. Another question we had was asking if you knew if aerobic exercise can affect vascular compliance.
Yeah, so yes, it seems like it does, at least over time, these things are related. I'm not sure of longitudinal studies that show the reversibility of stiffness. I think I saw a paper once that was finding that if you intervene early enough, you can reverse some arterial stiffness by having some aerobic intervention like earlier on before there's too much stiffening. But if you wait past a certain point, then it's harder to reverse, but you can still prevent more future changes. But yeah, I think it's not like super well studied yet, the following a longitudinal.
So another question that we had was about, like, were there specific types of exercise that you saw that were more effective or less effective in those studies about the impacts on the brain?
Yeah, definitely. So most of the studies have tried to replicate the earlier findings, which are usually using some aerobic interventions. And more recently, things have been moving like a little bit further off in directions of those, but that's definitely what most of the evidence is supportive of right now, is these like moderate aerobic activity. So this is like walking quickly or jogging for like 30 or 45 minutes a day seems to be really good. There's some evidence, growing evidence, that things like weightlifting are also useful, but there's less of that than these aerobic interventions.
All right. The next question we have is someone, I think, related to your field. So the question reads, okay. Clinically and pathologically, we distinguish between different types of dementia. For example, frontal temporal versus vascular versus LBD. Does cerebral vascular health seem to affect all of these equally, or is cerebral vascular health related to some types of dementia more than others?
Yeah, it's a really good question. It seems to be related to all of those types of dementias, but in very different ways. So like if you go into the nitty gritty, like molecular details of these interactions, there's definitely things that differ between these different types of dementias. And I'm really trying to focus this talk, or I was trying to focus this talk more on normal cognitive aging because I sort of see that as like, in some ways I see it as like a pathology, but like almost like a normal pathology rather than like these other sort of specific cycles that can change and affect different brain areas in different ways. But from a lot of what I've read, it seems like most of these things have some sort of vascular component that interacts with the underlying pathology. But it's not all the same.
And then the next question we have is more general, is what are you most excited about with this research going forward?
Yeah, so I'm really excited like this longitudinal component that I mentioned at the end. For me, it would be really cool if we could see, if our hypotheses are correct, that in my mind, if you can really like intervene really early on much future degrees of cerebrovascular decline with something like arterial stiffening, in some ways, it's something that we already sort of like, we have medications for that are useful to like decreasing blood pressure and some of the insults that can cause arterial stiffening to increase. If we knew that this was one of the main factors, we could really like intervene on it early or find ways to be more preventative in our approach for these things later in life. So I'm excited about the longitudinal aspects of this.
And then the next question we got was, when does the brain size decline start? At what age do you start seeing loss in brain volume?
Yeah, well, I started seeing loss in my own volume like right now. So I don't know. When we look at those, like the study I showed in this talk, I think showed some decline like around my age, but I think it's less typical. And I've seen other studies where it seems more flat until like the mid 40s, but I think it varies. It becomes easier to see the later you go, I guess. But at least for some individuals, it can happen earlier.
And then the last question that I got a couple of times was asking for those cognitive tests that you have. Is there like an answer key, or is there a way to like see how those work or explain the logic behind some of those?
Yeah, I guess I could explain the logic of them right now. Well, I don't want to explain the hard one because in case people want to keep trying it, I'll explain the easier one because not everyone got that right. Wait, I have to share again. Okay, so let's go back. So for these inductive reasoning tasks, you're really trying to find rules that reflect something that's happening. So like for this task, we see that this little circle here, it looks like it's moving one position over in a clockwise direction. So then for B here, it looks like it's moved one more over in the clockwise direction. So that's what we would expect to be the next answer. For this one, I won't give all of it, but you could think more about maybe like interactions between some of the boxes with each other. I guess I could give a hint that the direction of rules on this problem goes from top to bottom. But it's not like that's not in the instructions. Part of figuring out these tasks is trying to deal with that ambiguity of what is the direction even that the rules are occurring in. I assume that you're talking more about the logic of the inductive reasoning, whoever asked the question, but yeah, let me know if like one of the other tests was also confusing.
All right. Did anyone else have any other questions going forward? That's the end of the list that I got.
Okay, yeah, I guess I can put this up now then. And then I don't know, Danny, if people can stick around, maybe you could unmute them if they have more questions. And then I'll put the other reasoning task up for a little bit, just in case people want to keep working on it.
Yeah.
Okay.
Yeah, that sounds great. So thank you all for joining us. Please feel free to join us next month. It's it's the first Wednesday of every month. So next month it would be December 2nd. We'll have another talk, and then if anyone wants to stick around and ask more questions, I'll go ahead and have everyone unmute. But otherwise, thank you very much for joining us, and I hope to see you again in the future.
Yeah, thanks for coming, everyone, for the questions.